Chapter 5: Sensation and Perception. Perception begins with a stimulus from the environment, such as a sound or light, which stimulates one of the sense organs, like the ear or the eye. Sensation refers to the early processing that goes on. Our senses are our physiological capacities to provide input from the environment to our neurological system. What do all sensory systems have in common? They all have anatomical structures for collecting, filtering, and amplifying information from the environment. They also all have specialized receptor cells that transduce the environmental stimulus, such as sound waves or light waves, or chemicals, into neural signals. Neural signals are then passed along their specific neural pathways and are integrated in brain areas. Receptors share responses to stimuli and respond with a specific range and adaptation profile. With audition, receptors are located in the inner ear or the cochlea. The hair cells, specifically the tiny hair cells, are the primary receptors, and they convert sound energy to neural impulses and send them along to the primary auditory cortex. The anatomy of the ear includes the pinna, the external ear, the tympanic membrane or the eardrum, the middle ear, which is the hollow region containing the ossicles between the tympanic membrane and the cochlea, and the ossicles, the middle ear bones, as well as the inner ear, which includes the cochlea as well as the vestibular system. Hearing occurs via sound waves, which result from rapid changes in air pressure caused by vibrating objects. The vibrations formed by sound molecules in the ear canal project onto the tympanic membrane, which then controls the movement of the ossicles, the three ossicles, the malleus, incus, and stapes. The stapes then converts those movements to the cochlear cells, where the tiny hair cell receptor cells signal to the auditory nerve and the brain, the auditory cortex. The term tonotopic indicates that the cochlea and auditory cortex contain maps that are arranged according to the sound frequencies that most effectively stimulate the cells. In other words, there is a systematic representation of frequency. For example, neurons from neighboring receptors project to adjacent cells in the cortex. In a brief description of olfaction, odor molecules, or odorants, enter the nasal cavity and activate receptors embedded in the olfactory epithelium. There are over 1,000 types of receptors which can bind odorant molecules. The signal is then sent to neurons in the olfactory bulb known as the glomeruli. These pathways exit and form the olfactory nerve and extend to the primary olfactory cortex, and then project to the secondary olfactory area, as well as the hippocampus, thalamus, hypothalamus, and amygdala. The sense of olfaction is important in influencing autonomic behavior, attention, memory, and emotions. Next, we turn to vision. Visual information is contained in the wavelengths of light reflected from objects. The light passes through the cornea, pupil, and lens to the retinal receptor layers at the back of the eye. The image, first inverted, is focused on the retina, which contains 10 layers of neurons, with the deepest layer containing the photoreceptors, also known as the rods and cones. The rods and cones contain photopigments, which are sensitive to light and relay this information to the network of cells, eventually reaching ganglion cells and entering the optic nerve for delivery to the visual cortex. Rods and cones have several distinct features. Rods are most activated in low levels of light, whereas cones require intense light for activation. In rods, the pigment becomes depleted easily and takes a long time to replenish, whereas cones replenish photopigments more easily. Rods are more useful at night, and cones are more active during the day. Rods contain rhodopsin, whereas cones contain photoopsin. Rods are monochromatic, whereas cones contain 3 types: short, medium, and long wavelengths, or blue, green, and red. And they are densely packed in an area called the fovea. Photoreceptors provide for translation of the external stimulus of light into an internal neural signal that the brain can interpret. The short wavelength blue cones are maximally responsive to light with a wavelength of 430 nanometers. The peak sensitivities of the medium wavelength green and long wavelength red cones are shifted to longer wavelengths. White light, such as daylight, activates all 3 receptors because it contains all wavelengths. As discussed previously, the optic fibers from the temporal half of the retina project ipsilaterally, and the nasal fibers cross over at the optic chiasm. In this way, the input from each visual field is projected to the primary visual cortex in the contralateral hemisphere after the fibers synapse in the lateral geniculate nucleus. A small percentage of visual fibers of the optic nerve terminate in the superior colliculus for reflex function. Extensive processing occurs in the retina, where convergence of rods and cones onto ganglion cells occurs. There are 260 million photoreceptors and 2 million ganglion cells. Over 30 distinct cortical visual areas have been identified in the monkey, and evidence indicates that humans have even more. In this figure, each box demonstrates a distinct visual area of cortex as defined by physiology. The diagram shows how they are connected and how divergence and convergence exist across areas, allowing for multiple pathways of connection. Whereas all cortical processing begins in V1, the projections from 2 major processing streams occur. Neurons in the visual system represent space, as most neurons in the visual system respond only when the stimulus is in a specific point in space or its receptive field. There is an orderly relationship between receptive fields of neighboring cells, and external space is represented continuously with higher order brain regions. As there are over 30 cortical visual areas, study of visual perception is handled in a divide and conquer strategy. Each visual area provides its own limited analysis, and as information passes through the visual system, different areas elaborate and integrate the information from the primary visual cortex, or V1. Specialized regions in monkeys include MT, aka V5. It gets its name because it's located in the middle temporal lobe in the monkey. This is specifically sensitive to movement and direction, and also correlates to speed of motion. This graph shows speed tuning for a cell in MT in the monkey. In all conditions, the motion was in the optimal direction, and this cell responded most vigorously when the stimulus moved at 64 degrees per second. Researchers have employed neuroimaging techniques to describe the functional architecture of the human brain and have identified areas in the visual cortex that process color and motion specifically. Many scientists have used subtractive logic by factoring out the activation in a control condition from the activation in an experimental condition. This figure depicts an experiment using PET imaging with stimuli to identify regions involved in color and motion perception. With the control condition of the color experiment, participants passively viewed a collage of achromatic rectangles, all in gray tones. For the experimental condition, the gray patches were replaced by a variety of colors, and each color patch was matched in luminance to its corresponding gray patch. With this setup, neurons sensitive to luminance should be equally activated in control and experimental conditions. The colored stimulus should produce more activity in neural regions sensitive to chromatic information. These regions should be detected if the metabolic activity recorded when participants viewed the gray stimulus is subtracted from the activity recorded when participants viewed the color stimulus. The same logic was used to design the motion experiment. The black and white squares were moved every 5 seconds. Results indicated that in the color condition, the prominent activation was medial, in areas corresponding to human V4. In the motion condition, the activation was more lateral, in areas corresponding to human MT. Both stimuli produced significant activation in primary visual cortex when compared to a control condition in which there was no visual stimulation. Here we briefly discuss the relationship between sensation and perception, describing experiments that ask how activation in early sensory areas relates to our perceptual experience. Here we take information from sensory systems and use that to recognize objects and scenes. Early sensory processing identifies certain elementary features of an object, and that information is further integrated and refined on those pathways as those pathways move into secondary sensory areas. Further questions ask, at what point do we perceive an object? When does it become a percept and part of our experience? One way to study this is to trick our sensory processing systems with stimuli that cause us to form percepts that do not correspond to the true stimuli in the environment. By following the processing of such stimuli using fMRI, we can attempt to determine where in the processing stream the signals become derailed. In one study, it was asked if fMRI could be used to detect the neural fingerprints of unconscious perception. Participants viewed an annulus in which the lines were either oriented in only one direction, as is with the target, or both directions with the mask. Participants were asked to report which way the stimulus was oriented. When shown the stimulus for the 1/20th of a second, people had high accuracy in identifying orientation. However, if presented even faster, with the mask following the stimulus, performance dropped to chance levels. By using fMRI, the researchers found that the activity in V1 would distinguish which direction stimulus was presented, an effect that was lost in V2 and V3. As this example indicates, our primary sensory regions provide a representation that is closely linked to the physical stimulus, and our perceptual experience is more dependent on activity in second and association sensory areas. Note, though, that the examples base this argument on the fact that the absence of a perceptual experience was matched by the absence of detectable activity in secondary areas. We can also consider the flip side of the coin by asking what brain regions show activation patterns that correlated with illusory percepts. Stare at the enigma pattern shown here. After a few seconds, you should begin to see scintillating motion with the blue circles, an illusion created by their opposed orientation to the radial black and white lines. We know that moving patterns produce a strong response in V5, and this same area is activated during the illusion, as shown by PET and fMRI studies. Activity in V1 does not increase. This figure is from a study that looked at motion perception, with the goal to investigate if perception is more closely linked to secondary sensory areas. Monkeys were trained to make an eye movement, indicating the perceived direction of a patch of moving dots. The researchers recorded from a cell an area MT and passed a current through the electrode while the stimulus was present. The manipulation increased the probability that the monkey would report that the stimulus was moving in the cell's preferred direction, indicating that neuronal activity in the MT is important for motion perception. There is, of course, individual variation in perception, and researchers seek to identify these individual differences. One classic illusion study, devised by a German pioneer experimental psychologist, Hermann Ebbinghaus, asks participants to compare the size of the two circles seen here in the middle of the displays to the left and the right. Does one look larger than the other? By how much? Everyone sees the middle circle on the right as larger than the one on the left, but people vary considerably regarding how much larger they think the circle is. fMRI analysis revealed a negative correlation between the size of the illusion and the size of V1. The smaller the area of V1, the larger the perceived illusion. This may be because with a large visual cortex, each region of space has a better representation. In this illusion, the neighboring circles have less influence on the center circle when a larger V1 provides a higher resolution representation of space. Perception is in the brain anatomy of the beholder. When we refer to someone who's colorblind, we're usually describing a person who has inherited a gene that produces an abnormality in the photoreceptor system. Dichromats, people with only two photopigments, can be classified as red-green colorblind if they are missing the photopigment sensitive to either medium or long wavelengths, or blue-yellow colorblind if they are missing the short wavelength photopigment. Much rarer are disorders of color perception that arise from disturbances of the central nervous system. These disorders are called achromatopsia, where people see colors as different shades of gray. Other aspects of vision like depth and texture remain intact. This is consistently associated with lesions that encompass V4 and the region anterior to V4. In 1983, researchers at the Max Planck Institute in Munich reported a striking case of a woman who had incurred a selective loss of motion perception, or achinotopsia. This woman, MP, Her perception was akin to seeing the world as a series of snapshots. Rather than seeing things move continuously in space, she saw moving objects appear in one position and then another. CT scans revealed large, bilateral lesions involving temporoparietal cortices. With unilateral lesions, motion perception deficits are more subtle. Transcranial magnetic stimulation over human V5 area can produce transient deficits in motor perception. In one experiment, participants were asked to judge whether a stimulus moved up or down. The displays consisted of a patch of dots, only some of which moved in the target direction. The rest moved in random directions. Performance of the motion task was disrupted by stimulation over V5, creating a transient form of achinotopsia. Some areas of the brain, like the superior colliculus and superior temporal sulci, process information from more than one sensory modality and integrate the multimodal information to increase sensitivity and accuracy of perception. When the multisensory information is presented coincidentally in time and space, it leads to enhancement of the multisensory neural response.